Here we can see that the structure of chromosome is composed of DNA and histone protein. Initially, I told you that it is made up of two things. One is the DNA and one is the histone protein. And they makes the chromatin. One chromosome is made up of two identical chromatids. Two identical chromatids. So it talks about that chromosome where we have after semi-conservative replication. So once the DNA is condensed and it is not replicated now, it is only a chromatid, or you can say the chromosome itself. But some of the chromosome after replication join at their centromere, so they have two DNA molecule, each of which contains one DNA molecule. So one chromosome is made up of two DNA molecules, that is during the process of cell cycle. But if you're talking about the cell is not dividing, then each DNA just make of one chromatin. Chromosome can be seen only at the time of cell division. Otherwise, it remains a chromatin, a profuse mass. This is because each DNA molecule in the nucleus make an identical copy of itself, and two chromatids are held together by a narrow region called the centromere, forming a chromosome. Like here you can see the structure of chromosome. As initially I told, this is one of the uh, DNA molecule in the condensed form. This is another DNA molecule in the condensed form. So two DNA molecules are joined together at a narrow region, and this is called the centromere. The centromere is protected by kinetochore. So initially, if you just uncondense this DNA, it might like the mass, like here is an uncondensed mass of DNA. However, it also have the histone protein. Then it is called a chromatin. So when it's condensed, it become a chromosome. Right, so DNA is the molecule of inheritance, and it's made up of a series of genes. We have a lot of genes on the chromosomes. So a gene is also a segment of DNA. Each gene is one unit of inheritance, coding for one polypeptide. We already have discussed in the previous chapter. The fundamental subunit of chromatin is nucleosome. And this is something you have to study right now. Nucleosome. What is a nucleosome? So let's talk about the nucleosome. Here is the structure of nucleosome. This is one of the nucleosome. The nucleosome is made up of histone protein. Initially, I told you 8 histone protein, 4 on the upside, 4 on the lower side. Just I have told you in the previous lecture, and it is covered up by the DNA molecule. So what is a nucleosome? A nucleosome is a section of DNA that is wrapped around a core of protein. It's a section of DNA, not the entire DNA, only a part of the DNA, which is wrapped around the protein core. That protein core is called histone to form DNA protein complex called chromatin. So chromatin is a complex of DNA and histone protein. But if you talk about only one histone protein and the overlapping DNA, then it makes the nucleosome, which allow the DNA to be condensed into a smaller volume. When the chromatin is extended and viewed under a microscope, the structure resembles beads on a string. Beads on a string, exactly like the one we are using for as a tasbih. So that's the chromatin. Each of these tiny beads is called a nucleosome and has a diameter of approximately 11 nanometers. So it means, like I'm just giving you an example. This is one of the histone protein, and it's like a disc-shaped structure, and then it should be overlapped by the DNA molecule. However, the wrapping of DNA, like one of the wrap should be complete, and the other wrap should be more than a half, I mean 1.6 wrapping, and then it will be overlap on another histone protein. So again, here should be the overlapping on the DNA molecule. This DNA should be again wrapping the histone protein. Again, one wrap should be completed, the other should be more than, and then it makes a round on another one. So more than a half, and then it makes another round in the same way. So then it's make. So you can see that the middle core region, that are the protein, and the DNA is a double stranded structure. So let's me to make it like in this way. So it's like this structure. Now this entire structure is called a chromatin, which is made up of DNA overlapping on the histone protein. However, if you talk about only a single structure, like only this one with the histone protein and DNA, then this is called the nucleosome. So many nucleosomes makes the chromatin. So you can see that chromatin, nucleosome is the unit of, functional unit of the chromatin. The same story here. You can see here, this is the chromatin, this is the nucleosome, sorry. So here you can see, this is the nucleosome, these are the nucleosome. Many nucleosomes are grouped together. So some of the nucleosomes are quite condensed to make a heterochromatin, and some are quite open. These are ready for expression. Once we need the gene to be expressed, it should be in the euchromatin region. Euchromatin means active region. The gene are expressing here even during the process of replication. Some of the genes are expressing because they need some enzyme to be coded, and these enzymes are used in the DNA replication, like the I told you about the DNA polymerase, helicase, and topoisomerase, and so on. And this is the region which is the silent region. All the genes here are not related to those enzymes which are involved in the process of cell division. Of course, we have some more enzyme that will be switched on when the cell is specialized. After cell division, when the cell is going to be specialized, then at that time some of the genes are switched on. So these genes are sleeping right now. They are inactive in between here. So the condense, even in the chromatin, there is some condensed region, even in the chromatin. Once the chromatin entirely become condensed, it makes a chromosome. But even in the chromatin, we have some nucleosomes are so close together, which shows that these are the area where we have some silent gene. So nucleosome, what's nucleosome now? Which allow the DNA to be condensed into a smaller volume. Since DNA is a very, very longer molecule, so it must be wrap around the protein molecule to make it condense, and so that it can be occupied in a small unit of area. When the chromatin is extended and viewed under a microscope, the structure resembles bead on a string. Each of these tiny beads is called a nucleosome and has a diameter of 11 nanometer. I told you that this has a diameter of 11 nanometer. This has a distance, a diameter of 11 nanometer actually. Right. So each nucleosome is composed of a little less than 2 turns of DNA wrapped around a set of 8 protein called histone. It's like a small pieces of 8 protein to make a histone. And the DNA, one complete wrap and the other one is more than a half wrap. The chain of nucleosome is then compacted further and form highly organized complex of DNA and protein called a chromosome. Once the chain is completely compacted, like the entire chromosome become like the heterochromatin, then it makes the structure like a chromosome. Then we have two regions, condensed and uncondensed chromatin. This is the uncondensed, this is the condensed region of the chromatin. Chromatin exists in two forms, euchromatin and heterochromatin. Euchromatin is loosely coiled, like this one. You can see the loosely coiled, which can be seen during interphase having the active genes, whereas heterochromatin is tightly coiled in the chromosome, uh, as in the chromosome, seen at nuclear division, having mostly inactive region. So this region can only be apparent in the time of nuclear division because the genes should be compressed and they'll not be allowed to express this time, because at this time the cell is under the process of division, so we don't need the genes to be switched on. However, these are the genes which are always in a state of expression because they are related even to the process of cell division. In cell division we need enzymes and they can make the enzyme. In cell division we need the spindle fibers, the tubulin protein. So some of the genes should be for tubulin to produce the tubulin as a spindle fiber. So it means these genes are closely related to the enzymes and some other important protein which are involved in the process of cell division. Right. So Chromatin is at the most condensed form in chromosome at the metaphase stage of mitosis. Keep in mind that the complete and full condensed DNA is at the stage of metaphase. So means the process of condensation starts even in the interphase, I mean G2 phase. It remains continue. In prophase, the process of condensation still remain continue. Most of the chromosome become condensed. However, it's not the condensation is not completed now. The condensation is completed at the end of metaphase. So a metaphase chromosome is a fully condensed chromosome. This is very important to keep in mind. The main function of the chromosomes to separate the total information into two new cells, as all the information is tightly packed in it. Chromosomes also possess two features essential for the successful nuclear division. One is called centromeres and the other one is called telomeres. We talked about the centromere to some extent. However, we didn't talk about the telomere. We will discuss telomere in today's lecture. So what is centrosome first of all? It is the primary constriction, I mean the enfolded region in the two chromosomes of the condensed chromosomes, clearly visible with a light microscope. It can be seen with a light microscope, but once the chromosome becomes fully condensed. So you can't see the centromere when the DNA is not completely condensed. I mean, the centromere can only be seen at the later stage of prophase or at the early stage of metaphase where the chromosomes are fully condensed. It is the site of kinetochore assembly. Well, I told you what is now kinetochore is one of the protein just located on the centromere. So it is the site of kinetochore assembly. I mean, they are used to assemble the kinetochore on the chromosome, and is made up of highly repetitive heterochromatic DNA with non-histone protein. Keep in mind that in the centromere region there is no histone protein. There is no histone protein in the centromere region, and this is also non-coding part of the DNA. There's no gene to code some protein, and that region is made up of highly repetitive heterochromatic DNA. So heterochromatic means, I told you, that's the compacted DNA where the gene remains silent, but although there's no gene here, but still the DNA is only compressed with no histone protein. Then what is kinetochore? Kinetochore, each metaphase chromosome. Keep in mind that kinetochore can only be seen at the stage of metaphase. So each metaphase chromosome has two kinetochores at its centromere. You can see two kinetochore. One is there and the other one is here. And one on each chromatid. They are disc-shaped protein complexes intimately associated with centromeres and can only be seen by using an electron microscope. Very important to keep in mind that kinetochore cannot be seen using light microscope because there's a very tiny structure, disc which cover up the centromere. However, centromere can be seen by using light microscope. It is the site of assembly and disassembly of microtubule. You know, microtubule are the spindle fiber. So microtubule actually attached to the kinetochore, which is attached itself to the centromere. That's why we say that kinetochore is one of the site of assembly and disassembly of the spindle fiber. So spindle fiber are going to be attached here and also detached here. Once it's need to be attached for the pulling of the chromatids, they should be attached. And once we need to pull apart and separate the chromosome from one another and split the centromere, then they will involve in the disassembly of the spindle fiber. Right. Made up of multiple protein with trilaminar structure. Trilaminar means it's made up of three layers. If I'm just making like this part, if this is the kinetochore, so it's made up of three layers. The innermost layer is connected with the DNA. The outermost part is connected with the spindle fiber, and the inner part is the connective part which makes these two portions together. That's some of the structures trilaminar structure means. Right, so you can see here that the inner layer is in close contact with the condensed centromeric heterochromatin. I mean this part. Microtubules are attached to the outer layer, and the middle layer is less dense. So this part is less dense. Construction of kinetochore begins before nuclear division starts, during the S phase of the cell cycle, and are lost again afterward. This is about the kinetochore. Now, what is the function of kinetochore? I told you kinetochore is the site of attachment of spindle microtubule. Site of attachment of the spindle microtubule. Sometimes students remain confused and they say that spindle fibers are attached to the centromere. It's okay, it's not a mistake actually, but the real picture is like the centromere are covered by the kinetochore. So we can say that kinetochore is also a part of the centromere, and the spindle fiber not directly attached to the centromere, but rather they attach to the kinetochore on both sides of the chromosome. Is the site of attachment of the spindle microtubule and hence essential for accurate chromosome segregation. Segregation means separation. Unattached kinetochores also generate signal for mitotic checkpoint, which are S mitosis. That's very important point. Whenever we discuss about the checkpoint, I told you that there is an M checkpoint. M checkpoint is working at the end of metaphase, I told you, and it's a signaling protein which give the signal that all the chromosomes have been attached with the spindle fiber, and the spindle fiber is in accurate form, and now you can proceed the cell division, the nuclear division, and the chromosome will be split down. But if some of the spindle fiber, if some of the kinetochore remains empty and no spindle fiber are attached to them, so it is a signal for the checkpoint M, which generate a signal and says that the nuclear division should be arrested because the separation of chromosome is not equal, as some of the kinetochore are not attached with the spindle fiber. It means the nuclear division will be arrested at this stage, will be reversed to G0 phase. So means kinetochore is also one of the important part of the checkpoint, which is the M checkpoints. It provides signaling. It's also proteins that also control the signaling pathway. So it says unattached kinetochore also generate signal for mitotic checkpoint, which arrest mitosis until all kinetochore have correctly attached to the spindle microtubule. Otherwise, the chromosome will not be separated equally. Therefore, representing the major cell cycle control mechanism. This is one of the most important function of kinetochore. They play a very important role as a signaling control mechanism. So it's major cell cycle control mechanism, protecting against the loss of chromosome. The microtubule attached to a given kinetochore pull the kinetochore with the rest of its chromatid dragging behind. Mean it's just drag out the entire chromatid along with this kinetochore. So it's like that area will be splitting down, and this chromosome will be pulled away, and along with the kinetochore, the other will be pulled away from here, and the two chromatids will be separated from one another. And this is achieved by the shortening of the microtubule. I told you about the dimer story, the addition of dimer and removal of dimer. So shortening and lengthening of the spindle fiber actually involved to separate the two chromosomes from one another toward the both pole. Shortening of the microtubule occurs when the tubulin units are removed, I told you initially, from the microtubule. The poles of the spindle are where the centrosomes are located. I told you what are centrosomes, and centrosomes are the microtubule organizing center, a type of organelle having a condensed mass of cytoplasm located near the nucleus. So where the chromosomes are located at each pole. Let's talk about the centrosome as well. What is centrosome? A centrosome is an organelle located in the cytoplasm, usually close to the nucleus of the animal cell, and it acts as a microtubule organizing center, as I told you, and talks to the microtubule originating from centrosomes, moving toward the, not moving, growing, not moving, growing toward the kinetochore of each chromosome. Why we use the word growing? Because the dimer, I mean the tubulin protein, one by one is going to be attached to the chain and show the lengthening of the chain, I mean growing the chain. Right, for the construction of the spindle. It consists of two centrioles. Now, in one centrosome, we have two centriole, and centrioles are the major organelle which are involved in the spindle formation. But since they are inside the centrosome, and we say that centrosome have some more than 100 types of protein, maybe some other protein is involved in the spindle formation because it's the area of research. So it's suggested that it's more better to say that centrosomes are involved in the spindle formation rather than to say that centro, the centrioles are involved in the development of spindle fiber. So instead to say centrioles are involved, it's better to say centrosome because centrioles are inside the centrosome. Right. It consists of two centrioles that are oriented at right angle. Already you have been known, you have been told in chapter 1 to each other and are embedded in mass of amorphous material containing more than 100 different protein. So it's a mystery. Maybe the spindle fiber are arising from some other protein in the nucleosome. So any one of protein, either from the centriole or from some other protein, but since all these are located inside the centrosome, so it's obvious that centrosome is involved in the formation of spindle fiber. It is these protein that control production of the microtubule, not the centriole actually. So a centrosome is duplicated during S phase. Its doubling takes place in the S phase of the cell cycle. Just before mitosis, the two centrosomes move apart until they are on the opposite side of the nucleus. As mitosis proceeds, microtubules grows out from each centrosome with their plus ends growing toward the metaphase plate, equator of the spindle by adding tubulin monomers. So these cluster of microtubules are called spindle fibers. We already have discussed in mitosis in so much detail already in this process. So that's the way how the structure of chromosomes and different subcellular structures which are associated with the chromosomes are involved in the cell cycle. This is, I think it's very important to know about the idea of the structure of chromosome and the some other structure which are closely related with the chromosomes. Now, the other topic which we have to discuss right now, that is the telomere. So what is telomere? Let's discuss about the telomere. A telomere is actually a length of DNA, but it is located at the tip of the DNA molecule. Since a DNA molecule is a double-stranded structure, so each strand tip is sealed by a non-coding region of the DNA, and that is called the telomere. You can see the telomere here in this diagram. This is the telomere which is located on the entire DNA, and the end of the DNA is sealed by two telomeres. So the telomere is on one end of the DNA strand and the other as well. These are the telomeres. Similarly, on the other strand, we have the telomeres. Exactly in the same way, like you can see a plastic coat on your shoelaces. In the same, like one of the shoelaces, your DNA molecule, and the plastic coat exactly like the telomere. So what are telomere concerned? What they are doing actually? First of all, a telomere is a non-coding region of the DNA. It does not code any protein. Secondly, telomere uh, is the region where in one strand we have a greater number of the nucleotides cytosine, and in the other telomere, in their respective complementary strand of the DNA, the telomere contain guanine. So the guanine proportion in one strand, in the one telomere, and cytosine in the other strands are in rich. It means other nucleotide can also exist, but guanine and cytosine are in the higher proportion. Now, what they are actually doing, what the telomeres are concerned. At the time of replication of DNA, once the DNA molecule is going to be multiplied, like this is the DNA molecule, and I told you that during DNA semi-conservative replication, initially the hydrogen bonds will be broken down and the two strands will be separated from one another. So once the two strands are separated from one another after the breakage of the hydrogen bond, let me to show its telomere at the tip from the red one. These are the telomere after separation. And you know that the DNA polymerase then working on each template strand to grow the other chain of the newly formed DNA strand from the free nucleotide. I mean, if I'm let's to show, here is the enzyme. I'm sorry, here is the enzyme DNA polymerase who is working from 3 to 5. So if this is the 3 site and this is the 5th site of the DNA, then the enzyme is working on the DNA strand and looking the complementary coded, the chain is going to be formed. So the chain is going from 5 to 3, the new chain. As I told you in the previous chapter, the DNA replication will continue. If I'm giving you the example of this strand to be copied. Now, in this case, if the DNA molecule, the DNA polymerase is going to be arrive at this portion. So the telomere is like 6 to 7 or 8 base pairs long, and the sequence is like not coding any triplet code. They do not represent any particular amino acids. They are junk with one another. They are not in series of 3, but they are 4 or 5 or 6 are grouped together to make the non-coding part of the DNA. Now, in this case, I'm just giving you an example here of the one of the DNA polymerase. If the DNA polymerase arrive here, if it is arrive here, it means it should be the 3rd site, because RNA polymerase arrive on the 3 site and moving toward the 5th one, as I told you in the previous chapter. Now look at here, once it's moved ahead, you can see it cannot copy the first 3 nucleotide, as they are just under the enzyme, which is the DNA polymerase. So in front of the enzyme, we can see the TCC. So they will start coding of the nucleotide on the corresponding DNA template molecule. Once they make the new DNA molecule, it will start from the code. They can display the thiamine, they can display the cytosine, they can display this remaining 3 nucleotide, which is cytosine, which was cytosine. If you just focus on that area, you can see here is the cytosine and guanine. These 3 are missing as they are under the enzyme, and the enzyme cannot read them. They just start reading from the front, which is the thiamine, cytosine, and cytosine. So CCG, these 3 will not be read out by the enzyme DNA polymerase. So we have just to start from the TCC. So C, why I'm writing G again and again, C, and then the last one is the guanine. These 3 will be missing. Now the DNA remain continue until the DNA is now reached. The enzyme polymerase now reached here. Once it reached here, it again not move forward, so it again miss this part, half part of the telomere from both the end. So if the last end of the telomere, let me suppose, just give you a different color. If this is the telomere part, and they have a sequence like here, cytosine, guanine, adenine, thiamine, and cytosine. Maybe that the enzyme DNA polymerase reached only till here. So it will again miss 3 nucleotide. Which one? These 3 nucleotide will be missed because it cannot move forward. This is one of the problem with the DNA molecule, that the DNA one strand cannot be copied completely. The polymerase reach to certain level of the DNA and they just leave short part of the DNA on both the end on the telomere. That's the reason if the telomere was, if suppose the telomere was a functional part of the DNA, I mean it was, suppose if it were coding for the gene, then it will be a big loss. So a gene will be lost. So to ensure that no functional gene is missing while copying the DNA, we need some external and non-functional part to be added with the DNA, so that at least the DNA polymerase entirely copy the functional part of the DNA. If the telomere were not here, then the enzyme DNA polymerase will stop just few bases behind, and it might be a functional part of the gene to be lost. So to ensure that no functional gene is lost, we add some non-functional part, non-coding part of the DNA externally with the DNA. So the DNA polymerase will reach to the end, even they are not copying the entire DNA. But if something is missing, no problem. That is a non-functional part of the DNA. So it's play a very important role. It means that the new DNA molecule will be, which is copied, will be, it will be somewhat shorter in length than the original strand. So it means, I'm just giving you an example. If this is one of the DNA molecule and it has the telomere, this is the telomere. Once this DNA strand is copied in the next cycle, this DNA cycle will be like the functional part will be copied entirely, but some of the part of the telomere will be missing. That's why if the same cycle, if the same cell is divided again in the next cycle, again the functional part will be equal. However, the telomere part is still more lesser. And then in the next cycle, again, the functional part will be entirely copied each cycle, but the telomere will be further shortened, because the end of the DNA is not completely copied by the DNA polymerase. If this remain continue, a time will come when the entire telomere will be lost. Now there is no, the entire telomere is lost. Now, how many cycle this? If this is the first cycle, this is the original cell, and then the second cycle, third cycle, and fourth cycle. Now, in the fifth cycle, the DNA copy should be lesser in size, and maybe the part which is lost here, like this part which is lost, they may contain some functional gene which are very important for the survival of the cell. So if this gene were missing, the cell will not be survived and death of the cell will occur. It means it is a programmed death for the cell, that one cell after many division is finally died because its telomere is completely lost. And this is one of the reasons of our aging concept, why when we are going to older and older, our body becomes weaker, our bones become weaker, our skin becomes loops out, because we have decreased number of cells tremendously. Our number of cells in our body are going to be decreased due to the disintegration of large number of cells in our body, because the telomeres are ending with many cell cycle. So to sustain the telomeres on the DNA molecule, there is one of the enzyme which is called telomerase. Telomerase, involved to increase the length of the telomere again. Like at the end of the one cycle, some of the telomere has been lost. Now, once this cell is going to be divided in the next cycle, the telomerase try to again repair, again lengthen the telomere, so that no functional part of the gene is lost. However, the working of the telomerase and the time duration between the two cell cycle are not equal. The time duration between the two cell cycle is faster, it is lesser time, and the working of the DNA, the working of the of the telomerase takes more time. So before the telomerase to repair the telomere, make lengthen, the cells undergo division and further shorten, further shorten, further shorten, and at the end, some of the functional gene is lost, and the cells are going to be disintegrated. Like I just giving you the example of here, the telomeres here. This is one of the DNA molecule. You can see here, and in the DNA molecule, the shaded region are the telomeres here. So this is the shaded part of the telomere, and you can see the telomeres are also circled here. One of the telomere contains the sequence S, T, G, 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 C, A, C, G, G. So they have a rich proportion of guanine, 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 and the other telomere contains C, 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 C. Although they have G, T, and A, but the cytosine proportion is in higher. So this is one of the representation, and this is one of the identification of the telomere, that they have higher number of guanine and cytosine in their. Now these are the genes actually which I have shown in different colors. They have the gene. They have the gene. So once the DNA polymerase arrive here, let me shade it by some color. If arrive here, so this is not a good color. Let me do show it some dark color. If it arrives here, so it will lost the part of the telomere, but not lost the part of the gene, because it will settle down. Because for its settling, we need some part of the DNA. So if the enzyme DNA polymerase settle in such a part of the DNA which is a functional part, I mean which have a coding region, then that code will not be copied. So it is necessary that the DNA polymerase should be settled on the DNA strand in such a way that it is just behind for the code that should be copied. Otherwise, they will not copy the code which is just beneath the DNA polymerase. So that's why the DNA polymerase will copy the entire. However, it will reach half of that region. So again, it missed that part. And after each cycle, it just moved to a little area and leave the other part of the DNA. So that's why we say that telomere will not be copied. So this is one of the aging concept why we gets older and older, because our cell have the potential to be divided to certain level. So after 5 to 6 division, the cells are going to be disintegrated. The number of cells are decreasing day by day, and then our body is going to be weaker and weaker. So here some description now about the telomere here, telomere and its significance. The ends of the chromosomes are sealed by some extra non-coding region called telomere. Keep in mind, non-coding region called telomeres. They have been compared with the plastic tips on the end of the shoelaces. These DNA-based sequences are repeated many times, multiple repeat sequence. Repeat means they have same like guanine, 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 and cytosine, and so on. In telomeres, one strand of the DNA is rich in the base guanine, and the other strand is rich in the complementary base cytosine. Their main function is to ensure that when the DNA is replicated, the ends of the molecule are included in the replication and not left out. So if they were not there, some of the part of the DNA will be missed, because the enzyme needs some place to be arrive on the DNA molecule. So we need some more extra space for the enzyme to be where the enzyme ought to be settled on the DNA. It is exactly like a person is just cutting a branch of the tree. So if the person were to cut the entire branch of the tree, he must have to left some part where he sits to cut the branch on a head. But if he cuts the branch just from the bottom, the person will also fall down from the tree. So he must have some space to be left and then cut the piece. The same way, when the DNA polymerase is copying the DNA, it must have some place on the DNA where the enzyme to be settled, and that part is the telomere. Right. In telomeres, one strand of the DNA is rich in the okay, that's on. Their main function is to ensure that when the DNA is replicated, the ends of the molecule are included in the replication and not left out. The copying enzyme cannot run to the end of the DNA and complete the replication. It stops a little short of the end, just short at little part. So if part of the DNA is not copied, the piece of information, I mean the gene, should be lost. At each subsequent division, another small section of information would be lost. Eventually, the loss of vital genes would result in the cell death, because at each cycle, the DNA is going to be shortened and shortened, as at each division, the DNA polymerase not completely reach to the end of the DNA. Now, what's the solution? The solution is to make the DNA a bit longer by adding some more bases, although they are non-coding region, but just for the enzyme to be reached here. But allow the copying enzyme to complete the copying coded DNA. As long as extra bases are added during each cell cycle, no vital information will be lost and the cell will be able to continue dividing successfully. The enzyme that performs this role is called telomerase. So telomerase play a very important role. The main function, now what is the function of telomerase? The main function of telomerase is therefore to prevent the loss of genes during cell division and allow the continued replication of a cell. It has been shown that some cells do not top up their division. I mean, top up their telomere. They are not able to top up the telomere. So what will happen? They will be suddenly die. Like in the specialized cells, when the cells become specialized, they do not undergo cell division because their telomere has been lost. So, dividing successfully, the enzyme that performs this role is called telomerase. The main function of telomerase is therefore to prevent the loss of genes during cell division and to allow to continue replication of a cell. It has been shown that some cells do not top up their telomeres at each division. These tends to fully differentiate it. Why a cell is going to be differentiated? You know, I told you that there are two types of cells. One is the called stem cell, which has the potential to divide again and again. Because the enzyme telomerase is functioning there, they are adding extra non-base pairs, non-coding, non-coding base pairs, and allow the enzyme DNA polymerase to arrive again and again, so division will continue. But once the telomere is going to be ended and the enzyme does not work now, then that cell becomes specialized. It doesn't divide but start its function. With each division, their telomeres get a little shorter until the vital DNA is no longer protected and the cell dies. This could be one of the mechanisms of aging by which we grow older and die. This, of course, suggests that by somehow preventing the loss of telomeres, we might be able to slow down or even prevent the process of aging. It's very interesting. If we enable the DNA polymerase to lengthen the DNA by adding these telomeres more frequently and shortly with higher pace, then we can prevent the process of aging, or we can slow down the process of aging. So a person might be looking quite young from their physique, even at older age, because they have ample number of cells in their body and they show a strong appearance. So here again some key point of the significance of telomeres. And what are the telomeres? The ends of the chromatids in chromosomes are sealed with protective structures called telomeres. Keep in mind the ends of the chromatids in chromosomes. So the ends of the chromatid, how many chromatids we have? Two chromatids we have in the chromosomes. So the chromatids, there are two ends of the chromatids, and each chromatid is made up of two of the DNA molecule. They are made up of non-coding DNA that does not contain genes. That is made up of short base sequence that are repeated many times, multiple repeat sequence. In telomeres, one strand is rich in the base guanine and the other strand is rich in the complementary base cytosine. So at one end of the DNA, we have one telomere, and on the other strand of the DNA, we have the other telomere. So one DNA molecule carry two telomeres on both the ends. The main function of telomere is to ensure they have that the very ends of the DNA molecule are included in the DNA replication during mitosis. The copying enzyme responsible for the DNA replication is unable to run right to the very end of the DNA molecule and stops a little short of the end, as I told you. If this end part of the DNA molecule contained an important gene, that piece of genetic information would be lost during DNA replication. So, in each subsequent cell division, a little more genetic information would be lost. Telomere therefore act as a buffer region of non-essential DNA and ensure that no important coding section near the end of the DNA molecule are left out of the replication process. What does it mean? So and ensure what the telomere ensures that no important coding section near the ends of the DNA molecule are left out, because this is the non-essential part and they protect the essential part of the DNA. This ensures no genes are lost during cell division. I mean, the loss of vital genes can even result in the cell death and allows for controlled replication of the cell. To avoid the risk of losing genes, most cells have an enzyme called telomerase, as I told you, that adds additional bases at each end of the telomeres. Some cell, generally specialized cells, do not have telomeres to top up their telomeres, and therefore, after a certain number of cell division, the cell dies. This has been connected with the aging process, as I told you. So you can see the diagram here. This is the diagram here. We have this is the chromosome, and this is one of the chromatid, and this is another chromatid. So we have two chromatids here. So in one chromosome, we have four telomeres actually. One telomere on the top of both the ends of the chromatid, and here we have the telomere. So both the strands of the DNA make one telomere actually. So like one DNA molecule have two telomere, and two DNA molecule have four telomeres. So four telomeres here in one chromosomes, and one chromatid contain two telomeres. One chromatid is made up of one DNA molecule. So one DNA molecule or one chromatid contain two telomeres, and one chromosomes make up of four telomeres, and it contain two DNA molecules, and one DNA contain two telomeres. So this is the story of telomeres and their functions. In the next class, we will discuss some importance of mitosis, as what are the significance of mitosis, and then I will differentiate some differences between I'll point out some differences between the mitosis and meiosis as well, although meiosis is not in detail in this part of A level. However, we have it as a reference when we talk about the mitosis. So stay with us and see you in the next video.